Hello again, everyone. So now let us discuss the last exercise for this semester, the exercise 14 entitled The Degradation of Large Molecules.
What are carbohydrates? So, carbohydrates are the most common class of biochemical compounds. They include sugars and starch and are used to provide or store energy among other uses. Like most biochemical compounds, carbohydrates are built of small repeating units or monomers, which form bonds with each other to make larger molecules called polymers. In the case of carbohydrates, the small repeating units are known as monosaccharides, and each monosaccharide consists of 6 carbon atoms, as shown here in the picture. Early in decomposition, carbohydrates will be broken down by microorganisms. So the process begins with a breakdown of glycogen into glucose monomers. These sugar monomers can be completely decomposed to carbon dioxide and water or incompletely decomposed to various organic acids and alcohols or other oxygenated species such as ketones, aldehydes, esters, and ethers. Depending on the availability of oxygen in the environment, sugars will be decomposed by different organisms and into different products, although both routes may occur simultaneously. Under aerobic conditions, fungi and bacteria will decompose sugars into the following organic acids such as glucuronic acid, citric acid, and oxalic acid. And under anaerobic conditions, bacteria will decompose sugars into lactic acid, butyric acid, and acetic acid, which are collectively responsible for the acidic environment commonly associated with decomposing bodies. Other bacterial fermentation products include alcohols such as butyl and ethyl alcohol, acetone, and gases such as methane and hydrogen. The degradation of sugar molecules refers to the process by which the carbon chain of the sugar molecule is shortened by a single carbon. So simple carbohydrates like the glucose are fairly easy to metabolize and can be used to power up ATP or in the synthesis of proteins. More complex carbohydrates like starch or cellulose take a bit more effort as they need to be broken down into their component simple sugars before they can be processed. So to break them down, bacteria use a specific group of enzymes called cazymes, which stands for carbohydrate active enzymes, as enzymes are very specialized in the molecules that they break down. Different enzymes exist for different complex carbohydrates. The carbohydrate active enzyme is a sequence-based classification of enzymes that synthesize or break down saccharides, which originated with the seminal grouping of glycoside hydrolases. Carbohydrate active enzymes are the biological catalysts that synthesize and break down the multitude of complex carbohydrate structures found in nature. So different species from the genus Penicillium secrete a wide range of carbohydrate active enzymes or cazymes including beta-glucosidase that have several industrial applications. This glycosyl hydrolase hydrolyzes beta-link O glycosyl bond of various oligomeric saccharides or glycosides from cellulose. Some bacteria break down sugars by transporting the sucrose across the cell membrane and cleaves the glycosidic bond using an intracellular invertase. So extracellular glycosyl transferase polymerize the glucose molecules while liberating the fructose molecule so it is free to enter the bacterial cell. For instance, dietary sugars, starch, and fermentable carbohydrates are present in the diet and are in direct contact with plaque during eating and for some time afterwards. The breakdown of sugars is an important step that influences the plaque environment. Enzymes in bacteria and saliva break down sugars, polysaccharide, and disaccharides to monosaccharides. There are mechanisms by which oral streptococci hydrolyze or break down sucrose as shown in this picture. So extracellular invertase cleaves the energy-rich glycosidic bond between the glucose and fructose maltase. Then, the bacterial cell transports the sucrose across the cell membrane and cleaves the glycosidic bond using an intracellular invertase. The extracellular transferases polymerize the glucose molecule while liberating the fructose molecule so it is free to enter the bacterial cell, which streptococci are particularly proficient at this. Then, extracellular transferases polymerize the fructose while the glucose molecule is liberated, so it is free to enter the cell. And lastly, the salivary amylase cleaves the polysaccharides.
Good day everyone. Now let's talk about the principle behind the composition of lipids. Lipids may also be used as carbon sources by some microorganisms. Lipases produced by microorganisms split lipid into glycerol and fatty acid that can be used as carbon sources for energy and growth. As shown in Figure 1, triglycerides are a form of long-term energy storage in animals. They are made of glycerol and 3 fatty acids. Furthermore, microbial lipid toxins can induce membrane injuries and cell death. Many bacteria use exoenzymes such as lipase to hydrolytically decompose lipids. Two types of enzymes responsible for lipid degradation are lipases and lipoxygenases. Phospholipids compose the cell and organelle membranes of all organisms except the archaea. As shown in figure, phospholipid structure is similar to triglycerides except that one of the fatty acids is replaced by a phosphorylated head group. Triglycerides and phospholipids are broken down first by releasing fatty acid chains from the 3-carbon glycerol backbone. The reaction breaking down triglycerides are catalyzed by lipases, and those involving phospholipids are catalyzed by phospholipases. Those enzymes contribute to the virulence of certain microbes such as the bacterium Staphylococcus aureus and the fungus Cryptococcus neoformans. These microbes use phospholipases to destroy lipids and phospholipids in host cells and then use catabolic products for energy. Then, the resulting products of lipid catabolism, glycerol and fatty acids can be further degraded. Several types of lipids can be microbially degraded. Triglycerides are degraded by extracellular lipases, releasing fatty acids from glycerol backbone. Phospholipids are degraded by phospholipases, releasing fatty acids and phosphorylated head group from the glycerol backbone. Lipases and phospholipases act as virulence factors for certain pathogenic microbes. Fatty acids can be further degraded inside the cell through beta oxidation, which sequentially removes 2 carbon acetyl groups from the end of fatty acid chains. The ability of certain pathogens, like Mycobacterium tuberculosis, to degrade cholesterol contributes to their virulence. The side chains of cholesterol can be easily removed enzymatically, but degradation of the remaining fused rings is more problematic. The 4 fused rings are sequentially broken in a multi-step process facilitated by specific enzymes, and the resulting products, including pyruvate, can be further catabolized in the Krebs cycle. So the figure shows cholesterol side chain degradation in M. tuberculosis. Brackets indicate stages in the pathway requiring multiple enzymatic reactions. Pink text denote chemical name abbreviation of the catabolic intermediate.
Hello guys! Speaking right now is Sophie Rhea Maglangit, and I will be talking about the principles of the composition of proteins. Are you ready? Who among you here loves milk? If you do, give me a smile. Milk is a complex nutritional source that contains proteins, mainly casein, in an aqueous solution of lactose and minerals. Casein is a large milk protein incapable of permeating the plasma membrane of bacteria, and its presence is the reason why your favorite milk is white. Therefore, before casein can be used by some bacteria as their source of carbon and energy, it must be degraded into amino acids. So, how do bacteria use casein as their source of carbon if it cannot penetrate the plasma membrane? Bacteria accomplish this by secreting proteolytic enzymes that catalyze the hydrolysis of casein to yield amino acids. And this can be seen in your screen in figure 26.1, which are then, after yielding amino acids, they are transported into the cell and they are being catabolized. When milk is mixed with the plate count agar, the casein in the milk makes the agar cloudy, as you can see in your screen. Following inoculation of the plate count agar, bacteria that liberate proteases, just like casinase, will produce a zone of proteolysis. You can see it on the screen, the zone of proteolysis, in which it is a clear area surrounding the colony. So what does the clearing of the cloudy agar means? It means that it is a positive reaction and is the result of a hydrolytic reaction that yields soluble amino acids. In a negative reaction, you can see it in the figure 26.2b on the lower plate, that there is no protease activity and the medium surrounding the bacterial colony remains opaque. Shown in your screen are the example of proteolytic bacteria, which means that they can produce protease enzymes, which are enzymes that can break down peptide bonds in protein molecules. They are Pseudomonas, Bacillus, Clostridium, Proteus, Escherichia, Micrococcus, Microbacterium, Flavobacterium, Chryseobacterium, and many more.
So the materials that we will be using in this experiment includes 4 sections. So the first one is the bacterial cultures. So the bacterial cultures that we will be using includes Bacillus subtilis, Bacillus polymixa, Escherichia coli, Proteus vulgaris, Pseudomonas aeruginosa or Pseudomonas fluorescens, and an unknown bacterium. For the culture media, this includes litmus milk tube, nutrient gelatin tube, Shiras medium between 80 plate, and for the glasswares are dram vial number 8, sterile petri plates, test tubes, and the other apparatus includes alcohol lamp, disinfectant, ice cold water bath, test tube rack, and wire loop. So the first test that we will be conducting for the degradation of large molecules is the liquefaction of gelatin. So the procedure are the following. Step 1 is to inoculate the Bacillus subtilis, E. coli, and an unknown bacterium to 3 tubes of nutrient gelatin. Second, it will be incubated at 33 degrees Celsius for 47 to 72 hours. So after the incubation, it will be placed in an ice bath for 15 minutes. So if the medium in the ice bath is liquid, the gelatin will be liquefied. Otherwise, incubate for another week and observe again. So it usually takes 14 days for this experiment to be completed. So the expected results for the liquefaction of gelatin is that Bacillus subtilis yields a positive reaction for liquefaction, Escherichia coli yields a negative for liquefaction, and the unknown bacterium yields positive for liquefaction. So For the interpretation, if gelatin is hydrolyzed by the enzyme gelatinase, it does not gel when cooled but remains a liquid. Thus, it flows when the culture is tilted backward, like the right tube. And the negative tube on the left, notice that the solid gelatin does not flow when the tube is tilted. The principles of gelatin liquefaction: when boiled in water, the connective tissue collagen, which is a stringy, insoluble, and indigestible substance, changes into gelatin, which is a soluble mixture of polypeptides. So certain bacteria are able to hydrolyze gelatin by secreting a proteolytic enzyme called the gelatinase. The resulting amino acid can then be used as nutrients by the bacteria. Since hydrolyzed gelatin is no longer able to gel, it is a liquid. The ability of some bacteria to digest gelatin is an important characteristic in their differentiation. So this process takes place in two sequential reactions, and in the first reaction, gelatinases degrade gelatin to polypeptides. Then, the polypeptides are further converted into amino acids. The bacterial cells can then take up these amino acids and use them in their metabolic processes. For example, in our experiment, when grown on a gelatin medium, Bacillus subtilis causes liquefaction, whereas Escherichia coli does not. Gelatin hydrolysis can also be used to assess pathogenicity of certain bacteria. The production of gelatinase can often be correlated with the ability of a bacterium to break down tissue collagen and spread throughout the body of a host. So, gelatin liquefaction, or the formation of a liquid, can be tested for by stabbing nutrient gelatin deep tubes. Following incubation, the cultures are placed in a refrigerator or ice bath at 4 degrees Celsius until the bottom resolidifies. So if gelatin has been hydrolyzed, the medium will remain liquid after refrigeration. So if gelatin has not been hydrolyzed, it may require the nutrient gelatin up to 14-day incubation for positive results. So, in the clinical laboratory, gelatin hydrolysis is used to distinguish between the pathogenic Staphylococcus aureus, which is a gram-positive, and the non-pathogenic Staphylococcus epidermidis, which is slow negative. It can also be used to distinguish Listeria monocytogenes, a negative for gelatin hydrolysis, and one which causes bacterial meningitis from some species of Corynebacterium. Gram-positive, spore-forming, rod-shaped aerobic or anaerobic bacteria such as Bacillus anthracis, Bacillus cereus, Bacillus subtilis, Clostridium perfringens, and Clostridium tetani are also positive for gelatin hydrolysis. The test can also be used to differentiate genera of gelatinase-producing bacteria such as Serratia and Proteus from other members of the family Enterobacteriaceae.
Good day everyone. For the continuation of our report in exercise or activity 14, I'm going to tackle about the part of lipolytic activity.
So to test the lipolytic activity of the prepared bacterial culture, we will divide the agar plate into three sections. We will streak the Pseudomonas aeruginosa, the Bacillus polymixa, and the unknown bacterium to any of the sections of the agar plate. And then we will incubate the agar plate at 33 degrees Celsius for 48 to 72 hours. So after incubation, we will expect the following results reflected on the slide. So we will observe a wide zone of precipitation around the link of streak of lipolytic organism. Fats or triglycerides are composed of a glycerol molecule to which fatty acid molecules are covalently bonded through ester bonds. So triglycerides are primarily fat storage products in higher organisms such as animals. So some bacteria produce enzymes called the lipases that cleave the fatty acids from glycerol. So the fatty acids and glycerol can then be used for metabolic purposes such as synthesizing phospholipids for membrane construction or for catabolism to produce energy. So the decomposition of triglycerides and the breakdown of the fatty acids into short-chain volatile organic acid is the reason why butter or margarine becomes rancid. So fats are formed by ester linkages between three molecules of fatty acids and one molecule of glycerol. So simple fats are known as triglycerides or triglycerols. So these enzymes that break simple fats into its component fatty acids is, as we know, lipase. So many bacteria can be classified based on their ability to produce and secrete lipases. So a variety of simple fats can be used for this determination depending on the type of organism being tested. However, tributyrene is the simplest naturally occurring triglyceride and thus a common constituent of lipase testing media. So this constituent of the testing media is too large to enter the cell. So a lipase is released to break it down prior to cellular uptake. After hydrolysis, the glycerol can be converted to dihydroxyacetone phosphate and an intermediate of glycolysis. So the fatty acids are catabolized by a process called beta oxidation and may be converted into a variety of end products that can be used by the cell in energy production or other processes. So, for example, tributylene oil is a type of lipid called a triglyceride. Other lipase tests use different fat sources such as corn oil, olive oil, peanut oil, egg yolk, and soybean. So, in the experiment, the positive lipase test is the Pseudomonas aeruginosa, which shows a clear zone around the bacterial growth. So the negative lipase test is the unknown bacterium as well as the Bacillus polymixa, where the absence of a clear zone around the bacterial growth is not evident on the agar plate.
So this is the results in this figure. This is the result for screening of isolates. So in this table shows the activity that of every strain. So the double plus designates as the very clear hollow, and then a single plus designates clear hollow, and then negative no hollow form. So around colony and around microplate wells filled with supernatant. So the species that strains that shows double plus are the S247 and the S250, and then TC6, and then for the single plus is the SE17, and the rest are no forming hollows or observed no observed hollow formation. The second one is the result of the strain identification. So in figure 1, 16S RNA tree showing the phylogenetic relationship of neighbor joining method between strain TC6 and other known sequences of Notococcus sp. And the second figure shows the phase contrast micrograph of TC6. So, however, in this photo, further work on DNA to DNA homology and biochemical composition of cells, particularly polar lipid pattern, are under investigation of the determination of the species. So this slide shows the result of reduction of lipolytic activity during growth. This The first figure shows the effect of NaCl concentration on the growth and lipolytic activity of strain TC6 grown in 100 ml shake flask at 40 degrees Celsius and pH 7.5. The percent cellular proteins and the relative activity were defined as the percentage of the maximum value obtained respectively in the assay. Data are the average of two independent extreme AC conditions, NaCl3m and pH 8 and 40 degrees Celsius. The next figure shows the influence of pH on the growth and the lipolytic activity of TC6 at 40 degrees Celsius. The medium contained 3.5 m in ACl and has buffered with 100 mm tris HCl, as shown in the previous figure that I show in the phylogenetic decladogram that I shown in the last slide. The next figure is the temperature dependence of the specific growth rate of strain. So the TC6 at different NaCl concentrations at pH 7.5 results are average of two independent assays with standard deviations of less than 8%. And the next figure shows the combined effect of temperature and salt concentrations on the profile of lipolytic activity of strain TC6. The PNB, the A first, the PNB, and then the B is the PNP. The pH of the flask culture was 7.5, so results are average of two independent assays. So that's all.
The last test for this exercise is the litmus milk test. Milk is an excellent medium for the growth of microorganisms because it contains the milk protein casein, the sugar lactose, vitamins, minerals, and water. Litmus, a pH indicator, is incorporated in the medium for the detection of production of acid or alkali and oxidation reduction activities. Fermentation of lactose of milk with the production of lactic acid is demonstrated when the litmus turns from blue to pink as a result of acid production. The accumulation of acid acts on casein, resulting in formation of clot due to the precipitation of casein as calcium caseinate. With some organisms, the curd shrinks and whey is formed at the surface. Acid curd is identified by the clot, which remains immobile when the tube is inverted. Some bacteria produce renin enzyme that act on casein and in presence of calcium ions forms paracaseinate that is insoluble and is called rennet curd. Unlike acid curd, it is semi-solid clot that will flow slowly on tilting the tube. Some bacteria possess proteolytic enzyme caseinase and hydrolyze casein. Casein hydrolysis results in the release of large quantities of ammonia that makes the medium alkaline with foul smell. The litmus turns into a purplish blue. With further incubation, an opaque clearing of milk occurs as the casein is hydrolyzed to peptides and amino acids. The opaque liquid supernatant, which is whey-like appearance, turns brown in color. Additionally, some organisms reduce litmus, in which case the medium becomes colorless at the bottom of the tube. The media that we will be using is the litmus milk, and it is composed of powdered skim milk, litmus, sodium sulfite, and a pH of 6.8. The first step is to inoculate E. coli, Rictus vulgaris, Pseudomonas aeruginosa, Bacillus subtilis, and the unknown bacterium to the 5 litmus milk tubes. Then, incubate them at 33 degrees Celsius and observe every 24 to 48 hours. The expected results on this exercise is that E. coli, the unknown bacterium, and Bacillus subtilis tests positive, while Pseudomonas aeruginosa and Proteus vulgaris are negative. For it to be positive, it must show the following. If it is an acid pH, the color pink will turn to red. If it is an alkaline pH, the purple will turn to blue. In the reduction, it must be colored white. For the acid curd, there is a hard curd with clear supernatant or whey. For the rennet curd, there is a soft curd followed by peptonization. For the gas production, there must be bubbles in coagulated milk. For it to be negative, the color and the consistency remains the same.